welcome, say good afternoon or good morning to the participants in this webinar. It's a pleasure to be here. I'm going to give in the 35 minutes that I have, I want to give you a brief overview on arterial open wound and short time response treatment. And the goal there is to stop arterial hemorrhaging in 5 minutes, less than 5 minutes, by interfacing trauma site with an externally applied hemostasis agent, which is, as you'll see, is a nanostructured agent initially and then also at the end. And then I want to make a few comments on internal bleeding. There's really two categories here. Arterial open wound means it's something that would be treated by, as I show on the next slide, by compression directly. Internal bleeding, that's a much tougher problem, in which case you have to use targeted delivery for a hemostasis agent that will go to the trauma site without initiating any clotting or hemostasis before it gets to the clotting site. So I wanted to say a few words about, as I indicated, about the business of doing this with the inorganic and using the interfacial, using the targeted control of the bioprocess. Just a couple of words, because I don't have time to go into this in detail. This is a more recent work, and I wanted to give credit to particularly to my colleagues who are working in this area, Jim Morrissey and Stephanie Smith, who are at the Department of Biological Chemistry at the University of Michigan Medical School, and Hooman Rashidi, who's at University of California Davis School of Medicine, and Gary Brown, who's at the Burnham Institute for Medical Research. The project started on a collaborative research grant, which is from NIH and also from the Department of the Army in the United States, with between Professor Morrissey and myself at UCSB. And on the right-hand side, you see my colleagues, particularly my students who primarily started this, and they're Damien Cadella, who's taken what we initiated, and Professor Morrissey and I are scientific advisors for his new startup, Cayuga Biotech, which has been going on now for about 4 or 5 years and is still in its infancy, but it's making good progress. And Chi Quen, who is at the West Point, and Anna Masna, who are played two important roles in this project. So for the internal bleeding, that's indicated non-compressible wounds. And it's a situation where you have, say, blunt compact, like a bicycle accident, or it could be a car accident or whatever. But because internal bleeding, it can be it could be also from the venous bleeding, or it could be capillary, or it could be arterial. But the difficulty there and the challenge there, this is generally going to mean a longer term, and can lead to a situation where the body slows down and you get hypothermia, and you can also get the problems with the dilution, hemodilution, if other external fluids are used to replace the blood that's lost. And generally, it leads to, as shown on the left side, change the blood pH as well. And it's called the trauma triad of death, because once you get to coagulopathy, the blood's ability to clot is fatally implied. So it's impaired, rather. So the solution to that, a lot of it came from some inspiration from Professor Morrissey, who has been working on polyphosphates in human and bio living systems for many years. Your body uses polyphosphates in many ways, in the synthesis of bone, but also in the neural system as well. But every time you get a cut, the platelets extrude these polyphosphate oligomers, which are indicated here in the upper left, and they serve a purpose of really, through the common pathway of the blood clotting cascade, which I'll mention later, give you more detail later, activate or amplify the blood clotting process. The good thing about them is that, one, they don't initiate clotting. The 70-mer polyphosphates does not initiate clotting in normal blood, and if you coat a silicon nanoparticle with the 70-mer polyphosphates. It can be transported in, it's deliverable by cardiovascular without any side effects until it finds a trauma site where the blood clotting cascade is in process, at which times it switches on and activates and amplifies the process in a major way. And that's indicated it accelerates the clotting at the trauma site only. That's targeting that site, so wherever that bleeding may be. Promotes the clotting with only 33% plasma concentration, so it also addresses some of the hemodilution issues, and it promotes clotting even at 30 degrees C. Normal body temperature is 37 degrees C, which is where you have hypothermia. So it's a very promising. It still has. We're just getting into initiating the clinical trials. Has gone through the FDA and had approval in that respect, but there's still the clinical trials, and that's a long road ahead to get that accomplished. But we're very excited about this prospect. What I'm going to spend most of my time is talking about open wound arterial bleeding. And I'll give you the history about this. And this is going from the status of things in 2003. I was invited to work on this project at the beginning of the Iraq war military conflict. And at that particular time, this is the Pusateri Holcomb, the article you see at the bottom here, which was published in 2006, summarized it. Uncontrolled hemorrhage continues to be the leading cause of death due to military trauma, and the second leading cause of death in the civilian setting. And so this was, of course, before COVID-19, which has far surpassed those unfortunate records. But in the open wound arterial bleeding, the important thing here is you have your heart pumps about 4 to 5 liters of blood per minute, and you get mortality after you lose about 1.6 to 2 liters of blood loss, and that's usually in 3 to 5 minutes or less. So in an arterial bleeding, it has to be not treated very quickly. You have to do quick intervention to prevent death. And once you've prevented that, then of course you still have the problems with potential problems with hypothermia and hemodilution as well to go with that. So the strategy that we used to work on this project is indicated here. My research group did the material synthesis. The idea of thinking about different concepts and way we might be able to make a pragmatic solution to this, or arrive at a pragmatic solution to this problem. And we did in vitro blood testing at the University of California, Santa Barbara beginning in 2004. That we had a very close contact with the Naval Medical Research Center. When we found something that looked promising, the samples would go to what we created, would go to the Uniform University's center, hospital, medical research center, to validate our in vitro testing and the protocol we used for that. And if that looked promising, then it was immediately put into a swine model test, in which there'd be 8 to 10 animals, which would be tested for a single formulation. And they would do the in vivo testing. And if that looked good, then it would be packaged, go to Z-Medica for product development packaging. And in the process, there was a fast track at that time for FDA approval for the process. And then immediately sent out into the field, used for by the medical command, which I put astronaut, this is clinical trials in a very accelerated way, but it was and the feedback there came from the medical command people back to us. We communicated with them as well, so we knew that was the ultimate figure of merit test, of course, on how successful or unsuccessful we were. And that area, I just mentioned again the acknowledgments for that. And again, at UCSB, there's my students and the Stucky group, which was doing the therapeutic agent research, The hemostasis validation, as I mentioned earlier, was done then at the Office of Naval Research and the Naval Medical Research Center in Bethesda, Maryland, and also with particularly Hassan B. Alam, and who was at Mass General Hospital and Harvard Medical School, who also did that. And Richard McCarran and Michael Given were monitoring this project and gave good advice. And then if that worked, it went to the packaging and pharma validation, which was that under the medical guidance there was given by Giacomo Macedona, who was also at the University of Massachusetts Medical School. And then finally, after we had gone through all of this and we had arrived at a relatively promising solution, then there was the Institute of Surgical Research at Fort Sam Houston, which is Army based. The research I think I emphasized before was sponsored up until that point by the Office of Naval Research, but then they wanted to cross-check, of course, everything and therapeutically look at like the cytotoxicity and any longer range effects that maybe were not taken into account by the previous work that I had described. So anyway, the question is, I look at this as I'm an inorganic, bioinorganic, and I've worked on monitoring the growth of abalone shells and other thing else and trying to understand how biosystems control the assembly of inorganics in a very spectacular way, beautiful way. But, and out of that, actually, Angela Belcher was one of my graduate students who is now director of bioengineering at MIT, expanded on that and really developed phage display type approaches to create inorganic in a very nanostructured inorganics in all kinds in different ways, using bio direction to do that. But the other option was that you can also, then looking at it and based on some studies we made on living abalone, actually, we found that we could control the growth by the inorganic, so the inverse of that. So we could control the biosystem by the inorganic. So this was a rather, you have to understand, first of all, what I mean by systems here, and just a few words about that. So this is a picture of the blood clotting system. Blood clotting cascade system, and this was Damien Cudella, who was an undergraduate at Cornell and then came to work at my lab as a postgraduate student at UCSB. He worked with Jeff Varner, and then he did this, he was working on this blood clotting cascade system with Jeff Varner was, and Damien was also involved in that. So, but the important thing here is the blood clotting system is just not only coagulation, that's not our clotting, it's really an amazing type of integrated configuration in which there's initiation of the clotting, and then there's the clot formation itself. But in process, this, like many things, there's a yin and a yang to it. It's got, at the same time, you got to keep the coagulation local. And so there's anticoagulation factors involved in this, and that's important. That sort of regulates the whole process. There's about 220 species. There's 13 clotting factors have been identified, and this has been extensively studied by many blood biologists and medical scientists. And there's about 300 interactions. So it's somewhat on top of that, when you're thinking about applying it in the field, you also have to think about what about individuals who are either on blood thinners of warfarin or heparin, and the other parts that happen once the arterial bleeding is initiated. So looking at this, what our approach was basically, again, taking advantage of the scientific status of this, was looking at the various pathways of which the blood clotting works. And there's the intrinsic pathway, which is up in the upper left, and which is where, and this is both what's at the top here is really the clot initiation, and the extrinsic pathway, which is internal. They have two different, they involve activation and then different factors in the blood clotting factors in the blood clotting cascade. There's the tissue factor, factor 7, For example, in the internal and the external, the important one is factor 12. And the one that's commonly used for normal bleeding situations is really the extrinsic pathway. So that's the clot initiation. Then there's also the clot formation part of this pathway, which is shown here. The two intrinsic pathways, the two initial pathways, intrinsic and extrinsic, meet in what's called the common pathway, which is shown here. And this is really this is like the engineer. It controls both it feeds back into the intrinsic pathway clot initiation part of it, or I'm sorry, it feeds back into the clot initiation. And it also controls the formation of the clot, and it also controls the anti-clotting and the regulation part with various agents. And so all this is brought together. But basically, the important thing here is there are three pathways that are important. If you want to understand what the inorganic is doing, you have to understand which pathway it's going to use. I look at the inorganic, and having worked with materials for many years, they are also systems, and this define their system based on their properties. Heat release, the electrolyte. Calcium, incidentally, is designated as one of the clotting factors in the blood clotting cascades as factor 4. And it's, of course, a very important electrolyte and everything that goes on. And then there's the interacting surface potentials, the charge effects, and others that we've looked at. We examined all of these, and I don't have time to discuss all of them, but in greater and sometimes lesser detail, but that was very important. State of the art. Okay. Arterial bleeding had been a problem ever since military conflict was all through human history. And so I looked, when I first was thinking about this, I said, what's the state of the art? I thought surely it had been solved, but the fact is it had not. And the one that was the Navy went through and researched all the possibilities in 2002, 2003, and said, we like the idea of the dehydrated nanostructured zeolite as an hemostatic agent. And they did a test on this again with live animals and found they got 100% survival in the lethal model. And this was then packaged and sent to the military medical command use, and it was actually applied in the field. And it was applied just as shown here on the lower right as a powder on that. And the calcium 5A is it's aluminosilicate. It has one aluminum for one silica. These are tetrahedrally coordinated. So every place you have aluminum, it has a negative charge in the framework of the structure, which is shown on the next slide. And just a few words about, it's very quick. This is how this became, it was looked at and was thought it might be important. It came out of some work by an engineer named Maris, and engineer, not Maris, engineer. And he was working on air separation at Air Products, because if you totally dehydrate the zeolite calcium 5A, then you have a very highly charged framework here. In space, you have calcium ions, which are matching that charge, and you have very large electric fields, which are shown here as well. And that allows you to separate nitrogen from oxygen in dry air. And that's been a commercial process at Air Products and other places for many years. And the engineer was doing this actually, and this is described in that Nature article, cut himself when he was shaving one day, and he had a package of this totally dehydrated zeolite, and he put it on the bleeding site, and it stopped the bleeding. And he went ahead and he patented it. So the only problem with that is that the heat involved in that, the delta heat of hydration is 680 joules per gram when it's fully dehydrated. And the consequence of that is you get burns, second and third degree burns when you apply it to the skin. So this went in the field, but it's not it can stop the bleeding, but it also give you burns, second and third degree burns when you applied it. And so that was not an optimal choice. And so just a couple of other words on the nanostructure. The windows in the zeolite are 4.3 angstroms in diameter. And in addition to that, the so-called alpha cage, which is on the interior of this cube that you see here at the bottom, which is about 11.4, that's a bigger cage. And so is using these structures. So calcium is used. You can also do it with lithium, but calcium is, of course, more compatible And so the question is, there's several questions. Number one is, first of all, how do you stop the bleeding without the burns? And secondly, why does it work? Why is it calcium to liver? It could possibly be cauterization even, but there are some other issues involved in that. So that was where we started. And the bottom line is that we did this here, for example, with looking at the heating, controlled heating. A possible solution here is we could reduce the heat by using not using total dehydration, but just partial hydration. And so this is some visual thermal analysis that we were able to use, and we can monitor that and characterize the temperatures for given different amounts of water and picked out an optimal temperature for that. The in vitro testing, which is very common in the field and still used, and is basically looking at the shear elasticity and the dynamics of clot formation that takes place as the clot is forming. And the important parameters here are how long does it take for the clot, for the initiation of the clot? When does the clot start forming? How fast does it form, which is given by this slope of this called alpha here, as the elasticity and dynamics change. And then finally, and some evidence of what's called the clot strength. So, and you can do this as a function of temperature. And so the result was that when we partially controlled the heat in that way, in fact, you did get some improvement with the clotting time with increasing temperature. So that temperature that I showed you previously, the selector was used for that. I want to emphasize here, hypothermia, saying, well, it's not, that seems kind of trivial. It's not trivial as far as the body is concerned. The normal body temperature is 37 degrees centigrade. If the temperature, let's say, because you lost blood and the body starts to cool down and you get below 32.2 degrees, at that point you have 23% mortality. And if you're below that, you get 100% mortality. So the temperature is very important, and the time to clot formation is shown on the left. And so if your body has cooled down, then having something that warms up, it's not surprising that it might help as far as the clotting is concerned. There's other ways to modify this besides just changing the water, and we explored all of those. So I looked at, well, I shouldn't say all of them, we explored many of them, but basically you could replace some of the sodium and calcium ions that are balancing the charge in the nanostructured zeolite with silver. And if you put just a very small amount, 0.37% silver exchange, you could actually get a temperature control on that, so you didn't get the burns associated with it. And it had another effect, and that is it was antibiotic. And so we showed that this area of no growth was studied for various harmful bacteria that are infectious, that might cause infection. And that worked very well. You could also, we picked out here the zone clearance and the amount of silver that was used, and for some wounds and simple things that could use very small amounts, and the maximum you get was be about 0.4 silver atomic percent. The other part of it is the next step in this was always going in, if that improvement on things that would go to in vivo tests, and that was done by, again, the Uniform Services University Hospital in Bethesda. And I'd mentioned 8 to 10 pigs, and so this was looking usually controlled with the femoral artery or other. That's not the only one, but they basically looking at the bleeding. And so we looked at, I'm going to go through this quickly, but we looked at many other compositions and guessed substitution to see what would happen, and as a function of the mortality rate. And if you didn't do anything, it was 100%. And then the bag, we also at this point developed, instead of just spreading it on as powder, we also decided there's got to be a better way than that. So we came up with a tea bag delivery formulation. And the tea bag formulation gave us a 10% mortality, which is, 10% mortality rate, which is a good step forward. And again, this is again some looking at many different compositions and substitutions and finding out what would work. The important thing here, and particularly in the end, was not only how fast it clot forms, but also what's in the survival time. How long is it? What's its lifetime as far as being a functional therapeutic agent? And so that was also part of the study that was done in this publication that we did in 2006. And the fact that, again, the issue, of course, you can apply heat, but what does it do to the cells? And the T-bag optimum water calcium quick clot that we first worked on, it worked out quite well. But on the other hand, if you just, this is what you got to, with the regular sodium quick clot, and which is very damp, there was considerable thermal tissue damage, and we looked at it with other compositions as well. So where we stood at that particular point is summarized here as far as the clotting time and the in vitro studies were concerned. Basically, the optimum hydrated zeolite took about twice as long to initiate the clotting as did the totally dehydrated one, and it didn't clot quite as fast as shown by this quantity alpha, and the displacement was a little bit less, but it was definitely an improvement over over both the dry zeolite, we had no burns, and there was also improvement, of course, over not applying anything. And so that was led to deployment. It was packaged, and two materials, quick clot ACS plus TM and quick clot first response. Heat release is solved with some sacrifice of blood clotting efficiency. And then taking advantage of the silver as an antibiotic, there was also this, for example, the quick clot sports silver, which is sent out as well. So these are tested in the field, but they're also tested in civilian use. But the problem, I mean, the bottom line is, again, in the worst cases where you had a real femoral artery or real major artery bleeding. And this came back from the medical command, and the battlefield clinical conclusion says it's not good enough. You know, we're getting survivability is maybe 89 optimum in good days, maybe 90%, but it's more than that. It's also, once you would do it, how long does it keep the blood from, how long does the agent continue to provide hemostasis? So we went back again and looked at, said, oh, we got to go back to the fundamentals here, and let's make sure we understand the pathway. Now we can do that. You can do that by taking out different factors from the different pathways and applying them. So we took out factor 12 from the blood plasma, and for example, that controls the intrinsic pathway, which you see here for initiating the bleeding. And the result of that is shown here. Basically, if you don't have the factor 12, you don't get clotting. Even the totally dehydrated zeolite, nothing happens. Or I shouldn't say nothing happens, very little happens. So you get negligible decrease in the clotting time without it. And so it's pretty clear, okay, the activation, the way the inorganic was interfacing with the blood system, was interacting with the blood clotting system, was through the autoactivation of factor 12, is going through the what is called the intrinsic pathway. So thinking about that, we said, as I said, we were looking at other things. We did look at calcium uptake and delivery, and thinking about confinement effects and other aspects, but particularly we said, let's take a look at the surface potentials in the media. And SBF stands for simulated body fluid. You're going to measure these as is shown here. We did this with zeta potentials. Zeta potentials is electrical potential. That's what's called the slipping plane. That's the interface that separates mobile fluid from the fluid that's attached to the surface of the particle. And which is good for us, because and so for that, we start out, first we use, if you look up traditionally how you do zeta potentials, you use potassium chloride or potassium nitrate. And what you get with that is totally different than what you get if you use simulated body fluid or are with calcium chloride electrolyte. And so the bottom line here, this is simulated body fluid composition is here. And also, as I said, we use the calcium chloride electrolyte. But the bottom line here is, for example, if you look at silica, it tells you that silica below body pH is always going to have a very high negative charge and the zeta potential. Whereas if you look at zinc oxide, it's going to be positively charged, so that you can vary in a big way the charges on the inorganics and the zeta potential. And we mapped these out, and we then checked, and okay, how does this affect the clotting? And so in vitro, silica works very, very good. It's a great procoagulant. It depends on the particle size, nanometer size, and that's something that is important. And these are procoagulants at the isoelectric point. That's the point at which you going as a function of pH in the previous slide, where you go from a positive charge to a negative charge, negative to positive, and right there at where the zeta potential is 0, that's the isoelectric point. And so here's the, sorry about that. So here's the body pH 7.4, and so above that you have the anticoagulant, below that you have the procoagulant based on the isoelectric point. So this is a major success. It means we knew how to design an inorganic to make it procoagulant, anticoagulant, or procoagulant. And so then it's just the question of optimizing the material so that it fits all the requirements for the stability of it and its application. And hydroxyapatite, for example, has a very positive zeta potential. It's a very strong anticoagulant. It's primary function in your bone, and of course in the bone, blood cells are created inside. So so this is, and it's not surprising, but it is that. It's very strong. And then on the other hand, the silica is down here. Rate of clot formation is very fast, and the clotting time is very fast. I'm sorry, the rate of clot formation is very good and are strong, and the clotting time is very fast. And we could tune it. And so we took calcium hydroxyapatite, which had a positive zeta potential. We could create a silica doped or silica composition, mixed composition, and we could take it from being an anticoagulant to making it a very good coagulant. We wished that way. So we had lots of options in the materials. Okay, what was needed in the field was rapid response in vivo testing, lightweight, non-toxic, time and temperature stable. In the Death Valley, right recently, it was 130 degrees versus, of course, Antarctica, where it's minus 50, where it's, as far as storage of this material, or as it's carried by the individual. And active in the presence of anticoagulants, had to be dirt cheap and very cost effective and extensive validation. The solution of that, which we got, is shown here. And we tried some two-dimensional layered structures. And this is some, I call them layered clays, but they're not all clays. But anyway, the kaolin here had a very low clotting time, and it worked wet or dry. It didn't matter what it was. Here's the hydrated zeolite, which didn't clot well. And here's after it's clotted, after dehydrated, it works very well. And so the smectites and saponites turned out to be cytotoxic in a big way. This is just the zeta potential for kaolin, showing it at minus 31.7. But the smectites and saponites, they swellable. The reason that they were toxic was exfoliation. There's some animal testing on primates at San Antonio Army Medical Command Research Center that proved that, and so they were thrown out the window. They were not good. They didn't work. But this is really the important thing here. This slide here, the total blood loss, and this is for kaolin with what is called combat gauze here. Here's a dehydrated quicklime. And the total blood loss was half and with this kaolin as it was, or less, and the other. And furthermore, the total hemostasis time and a dehydrated we put it on, and how long was it doing, stopping the bleeding? With the dehydrated zeolite, it was just a very short time. With kaolin, it just kept plugging along, working almost forever. So that led to these compact gauze, which was formulated. And this, here's just some results. In vivo studies showed that kaolin-coated contact gauze controlled the bleeding in 100% injuries, liver, spleen, mesentery, and femoral arteries within 1 to 5 minutes from its application. The gauze did not become insaturated with blood. No other intervention was necessary. The controlled surgical gauze failed to control bleeding in all cases within 5 minutes from the application. And in several cases, because of persistent bleeding, the control gauze removed and the kaolin was then applied. And furthermore, besides the other hemostatic properties, it also had all the advantages of a regular surgical sponge. It was easy to use without required complicated training. It's pliable, could be introduced in deep cavities, even through small orifices. They're easily reaching distant bleeding points, and it was considered an exciting and promising new treatment choice for acute thromboembolic bleeding. And then there's testimonies for this. I've trained basic patients that come into the emergency medical with quick clot and position from early injury have had 100% survival rate, multiple saves in civil as well. And there's thousands of these that I could give you, but this was the back from people who actually used it. And so the Army liked it, and I got a nomination. I got recognition for it in terms of this. Like I mentioned, this funding was through the military and through the federal agencies, and so not allowed. The university patented, and that was I was not allowed to patent it myself. So and where we are right now, and this will be my last slide. It's still the hemostasis choice as of August 2019. And this is through the Committee on Tactical Combat Casualty Care. It analyzes this every year, and I haven't seen it for this 2020, but that's 10 years after first, or 11, almost 10 years after first went into service. 5 million units have been shipped to 5 branches, and many more than that. Actually, that's a low number. And there's no product-related adverse reactions reported. I have to say, I know one place where it's not, where I would qualify that statement, and that is applying it directly to the open cranial brain, and that's another story. But and so I think I'll stop here because of time, and I appreciate very much your listening, and I'll be happy to try to answer questions.